Hello friends, welcome to Science Place, and today we are going to know about history and introduction of microbiology. This is second part of this series, and let's see what is inside this. Going to know about how the theory of spontaneous generation was disproved and scientific contribution of Louis Pasteur and Robert Koch. So for theory of spontaneous generation, we have discussed a little in my last uh, lecture, that was lecture 1, but I thought a little more we should know about this, so I am repeating this theory again here. So what the theory of spontaneous generation says? This theory says that living things have originated from non-living matter spontaneously, and most of the people supported this theory in earlier olden days. Even the Aristotle, he believed that uh, the living things, living organisms or living bodies are made up of four elements, that are fire, earth, air and water. People believe that rodents can originate in moist grains, beetles can be originate from dust, and worms and frogs can be originated from mud, and rotten meat can give rise to worms. These were just simple observations when people see that like when they kept some bag of grains in some old dark area, and rodents obviously in the search of food, rodents will appear there, they may appear there, no? So this observation turned the people like the rodents originated spontaneously like that. But some people with great minds, they performed experiments to disprove the theory of spontaneous generation. There are few early experiments that were not that those scientists were not able to conclusively demonstrate that the theory of spontaneous generation is not okay. But later on, Louis Pasteur and John Tyndall finally disproved this theory. So let's see how were the experiments done by these scientists. So although there are many scientists, these four are very important, so I have taken these four scientists here. And the first one is Francesco Redi. What he did, he took three bottles like these, I presented here. One bottle was open. In all three bottles, there was a piece of meat, and one bottle was open. This one was open. The second one was sealed, and third one was sealed with a gauze cloth. That cloth was able to, that air can be passed from that gauze cloth, but flies could not pass that thing. So what happened, after putting these meat slices in these bottles, and he let these bottle kept for some time. After some time, he observed that in the open bottle, at the piece of meat, worms appeared, and in the sealed bottle there was nothing. But in the bottle that was partially sealed, you can see here, so maggots were appeared at the top of bottom where the smell could reach from this meat, but can't any nutrition can't go from this side to this side. And here these maggots laid down the eggs that appeared in the mature maggots in earlier in the later state. So this was very basic experiment. With this, he tried to prove that these maggots not originated in spontaneously, but because of these eggs were laid down. But people not believed in this theory, and later on, a beautiful experiment was performed by Lazzaro Spallanzani. What he did, he took this kind of flask, and some broth material was kept inside the flask that was having the extracts of plants and animals. And then this liquid broth was boiled properly to make this sterile. He performed the experiment in two arms, like one flask was kept open and the second broth was that was in the flask that was sealed properly and kept into the parallel. So after some time the opened flask developed the microorganisms while there was no any microbe in the sealed flask. So he said that because this air was not, he said ki air was having something that contaminated this broth, but people think in another way. They said ki air was drove off and the oxygen was drove off, that's why no organisms could grow in this flask. So this was also proof, but people not agreed completely. There was some yes no kind of thing. The next thing happened with the experiment of Louis Pasteur. He performed a beautiful experiment and presented in a France Academy of Science. That was a competition, and that competition was having the title to try by well performed experiment to throw new light on the question of spontaneous generation. So in this sponsored competition, he presented his experiment that was a beautifully designed experiment in which first he took a non-sterile liquid and poured it in a flask, and then the flask was heated. You can see here heated, and he gave a shape like this is the curve shape. Two cups were there, one is the upper side and one was the lower side. So after these curving, he then finally sterilized the liquid media also. So now this flask was having liquid media that was sterile and air can pass with this way. But because of this curve here, air, dust particles or any other microbe or small thing that is present in the air could not reach to this liquid broth. So despite of a long time, this broth was remain sterile. You can see here, है ना? The broth remained sterile. But when this flask was tilted and this broth was came here to touch the material that was present in the air, and then the broth came back to its original state, liquid become putrefied. So in this experiment, there was air present that you anyone could not say that because of absence of air could not anything grown, but this experiment finally proved that the material present in air that was like trapped in the dust fraction that was having something that originated this microbes inside this broth. So after this experiment, there is another evidence from produced by John Tyndall. He used this kind of chamber. That chamber was sealed properly, and he put some flask or tubes, you can see here, inside this chamber, and then he allowed the air to settle down. After some time, when until that point, these tubes or flask, whatever he put, they were sealed. But after settling of the air properly, that could be visualized with this some smart sort of window or pore hole kind of thing, that air settled properly. Because when we see the air in a dark box and uh, you know when you throw the light on that, because of Brownian movement, you can see the particles. But these particles also settle down with time if there is no movement of air, because this box was sealed properly. Air particles settle down after some time, and then he carefully removed the lids of these flasks, and after a long time he even after a long time he observed that these flasks were sterile. So he concluded that air could be sterilized by settling without any treatment that would prevent the vital force from acting. So after these primary and final experiments, this has been proved that there is no spontaneous generation exists. Life can be originated from life only. So thus the theory of spontaneous generation has been disproved. So whenever you are having some short note, you can go with these 4 experiments, and if you are having objective questions, I think you will be able to solve these. So let's move to the next section of this lecture, that is the contribution of Louis Pasteur. We have already discussed about how Louis Pasteur began to search in microbiology and how he contributed in the discovery of microorganisms, you know. So he was basically a chemist. He was a chemistry professor, but his interest lead to the discovery of many microorganisms and too many things in microbiology. So let's see what are the other contributions apart from that experiment that disproving the theory of spontaneous generation. So these are few points, like he discovered the mechanism of fermentation. He could see that the yeast cells are responsible for this process. Before this his observation, that was believed that fermentation is a non-living process, and he definitely performed the swan-neck flask experiment to disprove the theory of spontaneous generation. And then he because he wanted to sort out the uh, in the fermentation industry, because there were some contaminating microorganisms, and he wanted to sort out the things to make the wine and alcohol in a good way, so that uh, that industry can be maintained. So he discovered the method of pasteurization. Pasteurization is like heating of anything below 100 degrees Celsius to avoid or kill unwanted organisms. So initially he started to heat the wine at 56 degrees Celsius in presence of oxygen for 30 minutes, so that he could kill unwanted organisms. Okay, and then a very important thing, he discovered the vaccine of fowl cholera for chickens and rabies. This is the, very big contribution of him. And because he was experimenting a lot of on different microorganisms, and he performed many experiments on rabbits with the rabies thing. He used the rabbits, and he inoculated the rabies virus from a dog to rabbit, and then after so many cycles of infection, he obtained the dried spinal cord of rabbits that he used first time on a 9-year-old boy, Joseph Meis- Meister, who was bitten by a rabbit dog. And after a whole night thinking, because boy was definitely about to die, no cure was available. He was not a doctor, but he encouraged for this administration of vaccine what he produced, and he injected the boy 14 injections with uh, sequentially with a higher dose. He injected and immunized that boy, and finally that boy has survived. After this, his fame became too much in the world. From different people, uh, different part of the world, people started to came him for the vaccination of rabies. And that boy ultimately he uh, became the caretaker of Pasteur Tom in Paris. And with this, for the gratitude of this rabies vaccine production, ultimately the Pasteur Institute of Paris has been established, and he became the director of that institute in later stages. So these are few points about the Louis Pasteur. Uh, you can go with the short notes with this 4 points, and then after this Louis Pasteur contribution, we are going to know about Robert Koch. Before this, we can see what is germ theory of disease, that states that microorganisms can invade other living beings and cause disease. This concept was not accepted in mid-19th century. So after the experiment of Louis Pasteur, spontaneous generation discontinuation, this concept supported a lot of germ theory of disease, and in addition to this, Louis Pasteur also performed many experiments, but the first direct demonstration of the role of bacteria in causing disease came from Robert Koch. He was a German physician, and you can see how he worked in his laboratory. Robert Koch contributed a lot in the microbiology, and he developed the staining techniques. He developed the method to obtain pure culture. He discovered the anthrax is caused by a bacilli. He also discovered the causal agent of tuberculosis and cholera. And ultimately, he gave Koch postulate, and for the discovery of Mycobacterium tuberculosis bacilli in 1884, he got the Nobel Prize in Physiology or Medicine in 1905. So let's see his contribution in little bit detail. So what for the staining, he introduced the bedrock method of staining, that is using of methylene blue and Bismarck brown dye. These two dyes he was using to see the microorganisms. He was having his own microscope and camera. So he used to take fun by using just seeing the microorganisms by staining in his microscope. Then he also discovered about pure culture. So for pure culture, he performed a beautiful experiment. Uh, for this, he just first he took a boiled potato. Pure culture, because many people were using liquid culture, because but to study the microorganisms, we need pure culture. So he was trying to isolate pure bacterial culture. For that thing, he took a boiled potato, chopped this boiled potato in slices with a sterile knife, and then he inoculated the bacteria with the help of needle. And after that, he incubated this slice under a bale jar so that air could not contaminate that. And after some incubation period, he could isolate different type of colonies on the potato slide. But the thing was that potato was not supporting the growth of all kind of microorganisms. So he was looking for some alternative for that, or add-on to that. He used gelatin, but gelatin was having some issue, that gelatin melts at body temperature or even at incubator at 37 degrees Celsius, and even many microbes have the ability to degrade or liquefy the gelatin. So how he was some puzzled, and his lab associates were also puzzled, because one experiment goes wrong, so everyone tries in the lab to sort out that thing, so to solve this problem, this came the beautiful suggestion by Fanny Hayes. She was the wife of Walter Hayes. He was a physician and bacteriologist and working with the Robert Koch in his laboratory. So he discussed his wife that we are getting this kind of problem. Ye wo wo. So his wife suggested, why you are not using agar? So he said, what is that? She said, Ki, we use this for making jellies, jam. So as to solidify that. So he got surprised. Oh, okay, I will use that. And then he started, along with Robert Koch, definitely. Robert Koch just started to use agar as solidifying agent and liquid media, and that in that way they could find out the way to have the solid culture for isolation of bacteria using agar as the solidifying agent. The next discovery was the anthrax of bacilli by him. So how he established the relationship between bacillus and anthracis. This is really amazing experiment. Let's see this. So for this, he first took take a mice. Mice was healthy, but he also took a diseased mice, and some blood was extracted, syringed out from that mice and injected in this innocent mice. Now the mice was very afraid. Oh, what happened, Doctor Saab? What you did to me? I am feeling very bad. I am diseased. Then Robert Koch, after having the diseased symptoms appeared in the next mice, he took mice from this diseased mice and injected in another healthy mice. That mice also became ill. Then he further take out the blood from second mice and injected in third mice. As like that, he did this for up to 20 mice. After a series of experiments with 20 mice, he took out the spleen of mice, sliced it, and took a small piece of this spleen and kept in a beef serum. Beef serum grown with bacilli in culture as well as endospores. And then he put either bacilli or endospore in a healthy mice. Again, bichara healthy mice. He also became dead, diseased with anthrax. So you can see here that healthy mice get the disease from the unhealthy mice by using some blood, and this infection has been continued from mice to mice when one healthy mice was injected with blood from a diseased mice. So means that blood is having something, or some material in the body is having something. So when this piece of spleen was put in a beef serum and this bacilli has been grown, this was also able to produce disease in a healthy mice. By this way, first time he could link a specific microorganism with a specific disease, and by this way the final spontaneous generation was further rejected, and this experiment supported the germ theory of disease. Okay, and after this germ theory of disease, Robert Koch produces some Koch postulates. What these postulates were, but he not shown that postulate to at that point of time after the discovery of Mycobacterium tuberculosis. Then he you know publicly produced these Koch postulates in front of scientific community. So what were these postulate? Postulate means these are the criteria for providing the causal relationship between a microorganism and a specific disease. Means whenever you have to prove that a specific disease has been caused by a particular microorganism, these Koch postulates should be followed, fulfilled. And what these are? The first one is that the microorganism must be identified only in the diseased individual and not in the healthy individual. Mind it, only in the diseased individual and not in healthy individual. The next one is the microorganism should be isolated from the diseased individual and culture. First thing, it must be isolated from a disease, and second, it must be culturable. Then the cultured microorganism should cause disease when introduced to a healthy individual. And the next is the microbe should be isolated again from the experimental host and should be identical to the original organism. On the basis of this postulate, he discovered a deadly pathogen, that is Mycobacterium tuberculosis. How he discovered this? Let's see. So here you can see the first Koch postulate. What it says, the microorganism must be present in every case of disease but absent from healthy organism. So to fulfill this criteria, to observe this criteria, he developed a staining method of Mycobacterium tuberculosis. That was not literally the acid fast staining what we are doing nowadays, but that staining could able to identify the microorganism on slide. So when he took sample from the TB patient, he stained the organism and could see, yes, some bacilli are present, but these bacilli were absent in the healthy human. Then the second postulate says that the suspected microorganism must be isolated and grown in a pure culture. Okay, what second says? That it must be isolated and grown in pure culture. To fulfill this criteria, Mycobacterium tuberculosis was grown as pure culture on coagulated blood serum. Okay, so now this criteria has been also fulfilled because it could be grown on coagulated blood serum. The next one is the same disease must result when the isolated microorganism is inoculated into a healthy host. So for a healthy host, because human cannot be used, है ना? So he used a guinea pig. He injected the pure culture of Microbacterium tuberculosis in a guinea pig, and the bichara guinea pig died because of tuberculosis. And the next criteria is the same microorganism must be isolated again from the diseased host. So this died mice guinea pig, he isolated Mycobacterium tuberculosis from the dead guinea pig, and again he was able to culture the Microbiome tuberculosis on coagulated blood serum. So in brief, first see he seen the pathogen in the diseased host, then he was able to culture that organism. That culture was able to infect healthy animal, and after the disease he also he was also able to isolate the pathogen from this diseased animal. So these four postulates have been fulfilled and the Mycobacterium tuberculosis was discovered. And for this in 1905 he got a Nobel Prize. That was other thing in the first visit, in the first presentation, many people laughed on Robert Koch and he was not accepted. But later on when he evidenced this with the postulates, people believed in his, and now this is the Mycobacterium tuberculosis culture is the basic method of diagnosis. So thank you for watching this video. If you are liking this, please press the like button and subscribe the channel. Thank you so much. Bye bye. Take care.